네, 소개 감사합니다. 김상현이라고 합니다. 저는 오늘 Pp3 신항원을 표적으로 하는 T세포 치료제를 이용한 인간 고형암 치료란 내용으로 발표를 하도록 하겠습니다. 발표하기 앞서 이런 기회 주신 코센 관계자 여러분께 감사 말씀드립니다. 제 간단한 소개를 드리고 발표를 시작하겠습니다. 저는 암을 어떻게 이해하고 치료할 것인가라는 주제를 가지고 지금까지 계속 공부를 해오고 있습니다. 석사 과정은 서울대학교 서영준 교수님 연구실에서 암의 전반적인 기초에 대해서 공부를 했고, 그리고 이제 박사 과정 때 뭘 하는 게 좋을까 생각을 하다가 암의 유전적 특징을 알고 싶다는 생각으로 그 지놈 시퀀싱이나 마우스 지네틱스를 위주로 공부를 했고요. 이제 박사 후 과정을 선정을 할 때 그런 제가 박사과정 때 했던 것들을 어떻게 활용을 할까 생각을 하다가 스티븐 로젠버그 연구실에서 암의 유전적 특징을 표적으로 하는 면역치료제 개발 하는데 흥미를 느껴서 그쪽으로 공부를 시작하게 되었고, 이제 그 전과 다르게 임상실험이나 환자 샘플을 가지고 주로 연구를 진행하고 있습니다. 제가 나머지 발표는 영어로 준비를 해서 영어로 나머지를 진행하도록 하겠습니다. Today, I'll be talking about adoptive T-cell therapy targeting shared mutant Pp3 neoantigens. The goal of our laboratory is to develop an effective adoptive cell therapy targeting solid epithelial cancers, which account for 90% or greater of cancer death worldwide. It is well known that T cell can recognize and mediate tumor cell killing. One of the evidence comes from our experience with checkpoint inhibitor therapy. As shown in this left panel, T cell can recognize an antigen on the surface of tumor cell. However, tumor cell can develop a mechanism to evade this T cell based tumor cell killing. They can overexpress PD-L1, which can then interact with PD-1 on the T cell side to inhibit T cells activity against these tumor cells. If you can target this signaling axis between PD-1 and PD-L1 using antibody, then you can potentiate a T cell induced tumor cell death. Over the last several years, there were a number of FDA approvals for these few different classes of checkpoint inhibitors, starting first with the melanoma and across many different cancer types, suggesting this potentiating T cell through this PD-1, PD-L1 inhibition can be an effective therapy option for treating cancers. As Dr. Lee introduced in the previous presentation, one of the direct way of targeting cancer cells using T cells is a CAR therapy. Back in 2017, FDA approved the first gene-altering CAR therapy for B-cell leukemia. And there are two major classes. You can on the other side of this spectrum, there's a TCR, and TCR can recognize virtually any protein, and a tumor cell or any type of cell can process intracellular or cell surface protein to present antigen onto a molecule called MHC, or major histocompatibility complex, or in human is also known as HLA. So the question we have is, are there tumor cell specific antigens? So we came up with these two hypotheses. First is mutations in tumor cells can be immunogenic, and T cell can recognize the mutation-derived antigens, also known as neoantigens, via TCR. The second hypothesis is T cell targeting neoantigen can be adoptively transferred to patients with cancer to selectively eradicate the tumor cells. So to answer these questions, we developed a comprehensive screening method to identify tumor-specific neoantigens. So briefly, when a patient comes in, we resect the tumor and isolate DNA and RNA and sequence them to identify a somatic mutation in the tumor. We then prepare two types of material, which is a tandem mini-gene RNA that encode multiple different mutations original from this patient tumor. At the same time, we also synthesize a mutant peptides that contain a mutation in the middle of this peptide. By expressing these RNA or pulsing the peptides onto a APC or antigen presenting cells, such as dendritic cells, we then screen these mutation against the T cell directly from patient tumor or blood. We use markers such as 4-beta OX40, which are T cell activation markers, or we also measure interferon gamma or other cytokine levels using ELISA or ELISPA, which is a similar method as ELISA. Over the last several years, we screened 130 patients. And of those, we found neoantigens from 80% of those patients. And the median number of mutations that were identified was 123 mutations. So if you multiply these numbers, it was about 20,000 total mutations. And we screened about 3/4 of the mutations. In total, we found 210 neoantigens. So if you calculate the immunogenicity of this total of 15,000 mutations, it was about 1.3%. What we learned with this experience was all neoantigens were unique, except 2 patients that shared the same KRAS mutations. So this implies 2 things. First, either you completely individualize this type of treatment, meaning for a certain patient, you go from screening all these mutations to identify a neoantigen and develop a individualized T cell treatment, or alternatively, you can target shared mutations such as TP53, KRAS, or P53CA. My project focuses on TP53 mutation. One of the reasons is that the TP53 is the most commonly mutated gene across different cancer types. Here I'm listing all different types of cancer that have a P53 mutations. If you add all these up, it's about half of all cases have a mutation in P53, making it the most commonly mutated gene. Right, the question is then, are TP53 or P53 mutations immunogenic? Here I'm showing you one example from patient 4316, a metastatic colorectal cancer patient who contained a P53 C135Y mutation. We screened the TIL, so these are tumor infiltrating lymphocytes from this patient tumor. So we cut this big tumor into 24 different fragments and then individually grow them to maintain the T cell diversity. And here I'm showing you the interferon gamma secretion by these T cells. Here I'm showing the 5 different conditions. PP means a peptide pool that contain this P53 C135Y peptide. There's an irrelevant TMG or DMSO, which are negative control. And TMG is a tandem mini-gene that again contains P53 C135Y mutation. And PMA ionomycin is included as a positive control. As you can see here, as I highlighted in red, fragment 22 showed increase in interferon gamma secretion when co-cultured against the PC treated with the peptide pools or a tandem mini-gene. And here's the individual result. So in the negative conditions, you barely see dots. Positive condition like it looks like this. Peptide pools or PMA conditions were completely saturated because of too strong a signal. Because we tested tandem mutations or peptide pool contain multiple different mutations, we then individually tested these peptides that were included in the TMZ and peptide pools. We learned that canonical Pp3 mutation and two splice variant forms that again contain these mutations were positive for this screening. Compared to this negative control, this irrelevant peptide control or DMS control, we saw increase in 4-1BB T cell activation marker in fragment 22 that were tested. From these activated T cells, we isolated two different TCRs, one from here on the x-axis, I'm showing CD4. So one from CD4 positive activated T cells, the other from CD8 positive T cells. The top one is from the CD8 T cells, and here we reconstructed these two TCR into a retroviral vector, and then introduced them into a healthy donor peripheral blood lymphocytes, or PBL. And here we determined the specificity of this TCR by titrating these TCRs against wild type and mutant peptide. As we can see in both cases, this TCR shows a mutant specificity, although in the bottom we saw some wild type recognition at really high concentration of peptide. By repeating these processes for additional 77 patients, we identified a total of 39 TCRs. And here I'm listing the sources, those TCR, which is a patient number, and the type of Pp3 mutations. And the next column shows frequency of those mutations. So for example, this top one, this R175H mutant reactive TCR is can treat many numbers of patients because this mutation is one of the most frequent Pp3 mutation that's found in over 5% of all cancer patients. As I briefly mentioned, TCR can recognize an epitope or antigen only through HLA or MHC. And this TCR has HLA restriction of HLA-A0201, which is one of the most common HLA in the United States. So by combining these two numbers, we calculated a potential treatable number of patients. And by adding all the numbers up, we reach about 7.3%. These numbers are again for the entire patient population with solid cancers. Next, we tested these TCRs for their activity against tumor cells. First, we performed the in vitro experiment. Here, we're testing receptors against this Pp3 R175H mutation that I mentioned in the previous slide. Again, these TCRs have HLA-O2 restriction. And here again, I'm showing you the T cell activation marker 4-1BB level. These TCRs only increase the 4-1BB against a target tumor cell that have both HLA-O201 and Pp3 R175H mutation. As you can see here, if you have only a single component, such as these two cell lines or Pp3 mutation alone, T cells are not activated. But if you have both, you get significant T cell activation. Excuse me. Because this TCR3 showed a strong activity against these TYK and ovarian cancer cells, we further tested this TCR's activity using an in vivo model. This is a adoptive transfer model using NSG mice. we injected this TYK and new human ovarian cancer cells into these mice, waited about 2 to 3 weeks, and they injected T cells. These TYK and new ovarian cancer cells naturally express Pp3 R175H mutation and HLA-A02. These experiments were done in a doubly blinded manner. Tumors were measured twice a week, and PBLs were derived from healthy donors. So here's the result. Adoptive transfer of anti-Pp3 R175H TCR transduced PBL into NSG mice bearing TYK new ovarian cancer cell led to significant tumor growth inhibition. So here on the left, we are comparing 3 different groups. The first one in black is a irrelevant TCR that's not against this R175H mutation, and we are here testing 2 different dose levels for this R175H TCR. And as you can see here, treatment of using this uses R175H TCR at 2 different dose levels, all led to significant tumor growth inhibition and shrinkage of tumor. That also translated to improved overall survival of these mice. We further then tested this TCR in a clinical trial. One patient 4349, who's a 48-year-old female with metastatic breast cancer, with metastasis to the multiple organs such as lymph node, skin, pericardium, and the bone, received this treatment. This patient previously had progressed through 10 prior lines of chemo, radiation, and hormonal therapy. This patient was HLA-A02 positive. We sequenced her tumor and found this Pp3 R175H mutation. And we adoptively transferred 50 billion autologous peripheral blood lymphocyte PBLs transferred with this anti-Pp3 R175H TCR. And here's the result. Following the adoptive transfer of this R175H TCR transduced PBL, we saw a significant reduction in target tumor lesions. So starting from here on the left, as you can see here, pre-treatment, you can see the tumor deposit in the skin, and here, and also there was a tumor in the pericardium. Following a single treatment, the tumor significantly regressed. As you can see here, the skin and this lesion got much smaller. Also, this patient has a fungating tumor masses in her primary breast. And as you can see here, these are all tumor deposits which got completely resolved after this treatment. And in addition, we detected those infiltrating T cells that were transduced with this TCR. So using a technique called RNA scope, we mark these R175H TCR positive T cells that are infiltrating tumor and touching these tumor cells that were not seen in the pre-treatment samples. However, despite the initial response, patient 4349 progressed after 6 months of having a response. And when we sequenced this progressing tumor, we detected the loss of heterozygosity of this HLA. Excuse me. So on your left, this is a copy number analysis in the chromosomal level, and HLA is located in chromosome 6. So in the pre-treatment sample, we didn't see any loss of chromosome 6 here. However, in the progressing tumor, we detected a partial loss of a chromosome 6, which includes HLA. if you look more specifically at the HLA-A locus, pre-treatment samples, again, contain equivalent amount of HLA, both HLA-O2 and the other allele HLA-A24. Following the progression, the skin biopsy showed that a loss of this HLA-A02, while the other allele was maintained. To summarize, our data demonstrate Pp3 mutation can be immunogenic, and we identified a library of anti-mutant Pp3 TCR for potential adoptive cell therapies for a broad range of patients with solid cancer. This TCR recognized tumor cells in a Pp3 mutation and HLA-specific manner, both in vitro and in vivo. One patient with metastatic breast cancer received an adoptive cell therapy of autologous PBL transfused with the R175H TCR and had objective response for 6 months, but progressed with HLA loss of heterozygosity. Further studies are needed to evaluate the anti-tumor efficacy of anti-mutant Pp3 TCR in human clinical trials. So I'd like to acknowledge my mentor, Stephen Rosenberg, and the members of our lab who contributed this study. For the production of the T-cell infusion products for the patient treatment, we get help from tumor infiltrating lymphocyte production lab and also vector production facility at NCI. I'll take any questions. Thank you very much.